I'm a recent PhD graduate focusing my work on vaccine development from a machine learning perspective. So today we will tell you a little bit about our work back in February 2020, how we use computational tools to identify what should we put into a COVID vaccine. The key question we ask is, what should we include in a COVID-19 vaccine? The short answer is immunogenic components of virus. A component of virus can trigger immune response. A lot of us have heard about antibodies. Antibodies are proteins produced by B cells, directly bind to viruses to stop functions of viruses. One of them is entry of virus into human cells. And we call a part of viruses can be targeted by antibody B cell epitopes, as antibody is produced by B cells. But what if viruses have already entered the human cells? Fortunately, human has evolved a separate set of defense mechanisms based on T cells. Most of cells in our body have a machinery called MHC to sample a fraction of our protein and present on the surface of their own cell. And the corresponding T cell can identify any abnormal peptides, fragment of protein, present on the surface of cells and kill infected cells. So in short, COVID-19 vaccines should contain fragments which can trigger both B cell and the T cell response. And we call it B cell and the T cell epitopes. We ran these computational tools on all the possible proteins produced by COVID viruses. And the picture I'm showing here is the whole genome of the SARS-CoV-2, the virus caused COVID-19. One key protein I will be talking today is spike protein. It's this giant protein complex formed on the surface of viruses. It's so important because it interacts with human ACE2, which is the yellow protein I demonstrated here. Human ACE2 protein are commonly expressed on human lung cells, and the interaction between spike protein and the human ACE2 enable the entry of viruses. We scan the whole spike protein with SARS-CoV-2 computationally. The red region indicates the region the spike protein can be targeted by an antibody. As you can see, we identified two regions around 440 and 494 residues on the protein, the top candidates. Further, by collaborating with structural biologists, we identify these regions that actually sit between the interface of spike protein and the human ACE2 protein. So we have certain confidence if an antibody can target this region, it will have meaningful response in terms of viral infection. Antibody can potentially disrupt the binding between ACE2 and the spike protein to reduce the infection of human lung cells from viruses. Spike protein contains both T cell and B cell epitopes, and the anti-spike protein antibodies can potentially block the viral entry. We have a comprehensive list of candidates for either vaccine development and understanding T cell responses. We update our manuscript, and it's freely available on bioRxiv. Updates since February 2020 when we did this work. Globally, there are more than 130 vaccine candidates under different development phases, and most of them focus on spike proteins. If any of your non-science friends want to read more about COVID-19 vaccine design, specifically from AI angle, we wrote the opinion piece published by Brookings Institute. Right, so obviously, it only been 2-3 months, which is milliseconds if you measure in vaccine development world. Overall, people believe the top of spike protein, which I predicted, where the actions are. So which is ensuring, because top is where you interact with human ACE2.